In this video, we will discuss about the relationship of ER stress and p53 stabilization. So, what this figure conclude is, in overall, is that ER stress inhibit the stabilization of p53 proteins. So, we will start with the panel A that shows the Western blot that has been done. That is been carried out from the cells that are acquired from WY38 cells. These are the human diploid cells, or we can say first licensed human cells for the vaccine production. So what they have done is that they have used different kind of WI38 cells, that is cells that are treated with ADR or anti-cancer adriamycin drug. So what this drug does is these drugs will uh, this drug will leads to expression of p53 proteins. So when the Western blot is done after treatment of this anti-cancer drug adriamycin, and the cells were collected after administration of this anti-cancer drug adriamycin at different time intervals at 3 hours and at 6 hours. So at both the hours we can see that p53 can be production of p53 can be seen from the Western blot, or expression of p53 can be seen from the Western blot. However, when those cells were treated with TM, TM is basically introduce ER stress through inhibition of glycosylation. So this will cause stress within the ER, uh, within ER it is considered as ER is considered as the initiation of asparagine linked glycosylation. So inhibition of those glycosylation will lead to unstable structure accumulated within the ER that will cause ER stress. So when the ER stress is introduced through this TM or uh, tunicmacin, tunicmacin, then the production of or expression of p53 is reduced. And when thepsigargin is introduced to those cells, so the p53 is reduced further. Thepsigargin is cause disruption in the calcium uh, calcium circa cycle that will cause ER stress within the cells. So when the ER stress is introduced, the p53 expression is reduced, or we can say the p53 is not been stabilized in case of ER stress. And when same experiment is done for elongated period of time, we can see that same experiment is done with the elongated period of time, and also this time they have also introduced, and this time instead of using this uh, we can say this time they have also introduced this uh, actinomycin D. So what this actinomycin D does is this actinomycin D will stabilize the p53 proteins, uh, as in case as normally p53 proteins are stabilized by phosphorylation at serine at 15 and 20. I'm not sure if these two positions are accurate or not, but it's phosphorylation of serines at two different position in the p53 will stabilize the p53 proteins. So one of the hypotheses could be that the ER stress will disrupt the phosphorylation process that might lead to unstabilization. So in order to test that hypothesis, they have researchers or have introduced or this actinomycin D. It will stabilizes the serine at those positions, or serine at 15 and 20 positions. And so in that case, if phosphorylation, if ER state disrupt the phosphorylation, then in this condition the p53 expression will does not get affected, as those serines get stabilized by this actinomycin D. However, in the results we can see that there is a decrease in the p53 uh, p53 expression that can lead to the point that ER stress does not disrupt the phosphorylation of p53. Now they have tested this ER stress relation with p53 on different cell line, that is Hackett 113 cell lines, in which this p53 is introduced through this FU, or we can say 5 through the uh, this 5 fluorouracils. So cells that are treated with 5 fluorouracils will show p53 expression. However, when they are also treated with this TM and thepsigargin, we can see that the expression of this p53 is reduced compared to the this cells that are treated with 5 fluoro cells. And these things we further validated with the cell cycle assay. We can see that one of the function of p53 is the is to repair the cell DNA damage within the cells, and if the DNA damage within the cells is unrepairable, then it will uh, trigger the apoptosis. And in case of cells treated with fluorouracil, then p53 will triggers apoptosis, as the cell DNA has DNA damage is beyond the threshold level. So when this thing is tested using the cell cycle assay, we can see that in the control the number of cell deaths are less, or sub G1 levels, cells in the sub G1 levels are less, around 3.1, 3.2 and 5.5, and these are cells without treated with this 5 fluorouracils. So now at this time the expression of p53 is at the normal level as in the control. However, when the cells are treated with 5 fluorouracils, we can see that the cells in the sub G1 levels, sub G1 cycles is more, and when they are treated with thepsigargin and tunicmacin, we can see that number of cells in the sub G1 level reduced compared to the cells that are control cells that are treated with 5 fluorouracils. And these first two lanes in the panel D is done, experiment is done on p53 positive cells, or Hackett cells that have p53 genes as the wild type p53 genes. And the next experiment is done on p53 knockout Hackett 116 cells. So in this we can see that there is not much change that is observed in the sub G1 levels, either in the control and also in the cell lines that are treated with 5 fluorouracils, as even though the 5 fluorouracils will promote the expression of p53, since the p53 is not available, or p53 gene is uh, not available as the wild type p53 gene, or there is a mutation in the p53 gene, or we can say there is a knockout of p53. Because of that, there is no trigger of cell death because of p53 observes. So this thing will supplements, or this experiment will supplements, or we can support their point that ER stress inhibit p53 stabilization. And the final experiment is done on the colony formation, as we can see that in the first figure, or the top figure of the panel E, the colony formation is uh, we can say high in case of the top cells, as there is no trigger for the expression of p53. However, when the cells are treated with 5 fluorouracils, we can see that the cell population reduced dramatically. However, when the cells are treated with thepsigargin and tunomacin, cell population is more compared to the control cells that are treated with 5-fluorouracils. The quantitative value are represented here. We can see that in the control cell that are treated with 5-fluorouracils, the cell population survival is around 6.1%, whereas in case of other where the ER stress is introduced artificially, cell population is increased, that is 22.6% or 20.6%. However, when the same experiment is done with p53 knockout cells, we can see that there is uh, cell population is high when the cells are not treated with 5-fluorouracils. However, cell population is decreased. However, remains same in all the three condition. Uh, that is when the cells are under ER stress or the cells are not under ER stress. So this shows that the p53 stabilize p53 uh, stabilization get inhibited by the ER stress. By if we if we combine both the figures and make a conclusion, then one of the conclusion would be that ER stress inhibit p53 stabilization, as can be seen here, and there is no changes in the panel second figure of the panel E. The reduced in cell population in this case could be because of other reason, and that is common in all the three cases. So I hope you have understood how the author has uh, made his point or made their point that he has stress inhibited p53 stabilization. If you need to understand this thing in details and go through other part of this paper, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.